Hey everyone, in this video I'm going to talk about altering DNA. Now the first part of this is going to be talking about mutations. And mutations is something that can happen in both prokaryotic and eukaryotic cells. The second part of this is going to be talking about prokaryotes and some unique features that they have for altering their DNA. Let's get started. So mutations are random changes in the DNA, and they can be for entire chromosomes, such as what we talked about with non-disjunction back in genetics, or they could be at the nucleotide level. And the types of nucleotide level mutations include two types. The first is called a point mutation. This is a single nucleotide change. You may remember these called substitutions. And the results of these point mutations are three different possibilities. The first is called a nonsense mutation. With the nonsense mutation, the codon that is supposed to be reading for an actual amino acid now accidentally codes for stop. So you would see these types of mutations where a protein has been cut short and is non-functional. The next type is called a missense. In a missense mutation, the codon now codes for a different amino acid. You would know it's a missense because every amino acid before and after the mutation is identical and only one amino acid has changed. The last type of mutation is called a silent mutation. In a silent mutation, even though there's been a change to the nucleotide themselves, like the A, T, C, G, the same amino acid is going to be encoded, so you wouldn't notice any difference in the amino acid chain. Now, sometimes this is referred to as wobble. So wobble is when the last letter of the codon can be changed without affecting the amino acid itself. So there's some redundancy, there's some repeats of what these codons code for. In case there's a mutation, then you shouldn't have that devastating of effect if it's on the last letter of the codon. The second type of mutation is what's called a frame shift. And this is caused by inserting or deleting one of the nucleotides in either the DNA or in the mRNA. Now you'll know you're looking at a frame shift mutation because everything after the mutation is completely different than the original. There are some ways that a frame shift mutation, in other words, the insertion or deletion of that nucleotide, can cause a nonsense mutation. So just be aware that that's possible. But typically in frame shift, what you're going to see is every single amino acid after the mutation point is now different. Next, we're going to talk about prokaryotes. And with prokaryotes, in addition to having the ability to change their DNA through these mutations, prokaryotes can actually force DNA changes by doing or incorporating other pieces of DNA. There's several ways to do it. So for prokaryotes, the four different ways that prokaryotic DNA can change, aside from mutations, are conjugation, transformation, transduction, and transposons. In conjugation, bacteria are actually able to exchange genetic information with another bacterial cell. This would be akin to sexual reproduction, where there is an exchange of gametes. The second way that prokaryotes can change their DNA is a process called transformation. In transformation, a single bacteria can actually take up plasmids. Now, plasmids are pieces of DNA that are just lying around. Now, understand that there is DNA everywhere. There are dead cells everywhere, and as these cells break down, their DNA is also released into the environment, and bacteria can actually take these plasmids, these external DNA, and incorporate them into their own DNA. And then it's part of their genome. And every time that cell replicates, it replicates the plasmid as well. If the bacteria picks up a plasmid that offers some type of benefit, then that bacterial cell is going to be able to divide and be more successful at passing on that genetic information to the next generation. If the bacteria takes up a plasmid that is harmful, in other words, that it actually, the DNA that they pick up creates more problems than it solves, or if it doesn't do anything, like it doesn't help the bacteria, then the bacteria that take up those plasmids, those external pieces of DNA, they aren't going to benefit from it. The next process is called transduction. Transduction is when a virus causes the change in DNA for these prokaryotes, for a bacteria. So these viruses are called bacteriophage because they specifically target bacteria. We're going to be talking about this process of transduction and transformation in much more detail in our biotechnology unit later on this year. The last method that prokaryotes can use to change, or that prokaryotic DNA can change, are called transposons. And for transposons, segments of DNA are shifted to another location. So an entire segment of DNA may slide down to another area of the DNA, and this is going to ultimately cause a change in the proteins that are produced. The goal of altering the DNA of organisms is to increase the amount of genetic variation found in the cells. This genetic variation is going to be the source of evolution, as different phenotypes determined by the DNA of the organisms themselves may have favorable or unfavorable interactions with the environment. I hope this video was helpful. Thanks for watching. Bye.